Hello everybody, we are looking at question number 18 from GATE Biotechnology 2012 paper. So the question is, identify the statement that is not applicable to an enzyme catalyzed reaction. So the options are option A, enzyme catalysis involves propinquity effects. Option B, the binding of the substrate to the active site causes a strain in the substrate. And option C, enzymes do not accelerate the rate of reverse reaction. And option D, enzyme catalysis involves acid-base chemistry. So to answer the question, let's learn about enzyme catalysis. So first of all, enzymes are a type of biocatalysts used to modify the rates of reactions in plants and animals. So enzyme catalysis is the process by which there is an increase in the rate of the reaction through an enzyme. So it is important because most of the biochemical reactions have meager rates of reaction when uncatalyzed. So for a reaction to occur, the reactant molecules should have sufficient energy to cross the energy barrier. So it's called the activation energy. So activation energy is defined as the minimum amount of extra energy required by a reacting molecule to get converted into the product. It can also be described as the minimum amount of energy needed to activate or energize molecules or atoms so that they undergo a chemical reaction or transformation. So in simple terms, all the reactant molecules, they already have energy themselves, but they need some extra energy so that they could collide with each other to react and form the product. So this extra energy that's required by the reactants, that is the substrates, is called activation energy or free energy. So if the activation energy is so high, it means that the molecules, the reactant molecules, they need more extra energy for them to react. So that is that might not be possible in many cases. So this leads to the reactant molecules not reacting, or only very few of those molecules could be having a high activation energy for them to form a product. So what enzymes do is they interact with the reactant or the substrate, and they help to reduce this activation energy. That is, they reduce the minimum energy required by the reactants. So let's say we look at this purple line. So now the reactants have the energy up to this dotted line. So it needs this much energy. So it needs more energy for it to be active. But by interacting with the enzyme, it needs only half the amount of the activation energy that it needed without the enzyme. So in this case, it's possible that more reactant molecules in a given reaction mixture could have this extra energy, and so we just have more reactants to collide with each other and form the product. So like I said, all the molecules possess energy, only few have enough energy higher than the activation energy. So enzymes in the native conformation, they form complexes with the substrate and reduce their activation energy. So the 3D conformation of enzymes have an active site where the substrate bind and is acted upon. So the active site generally has two sites. So one is the binding site where the substrate binds, and one is the catalytic site where the enzyme catalysis takes place. So the enzyme is generally flexible. So the flexible enzyme changes its shape throughout a process called induced fit when the substrate binds to it, forming an enzyme substrate complex. So sometimes the enzymes also need cofactors like other molecules to help them in the reaction. So the apoenzyme, which is just the enzyme and its domain that binds to the substrate. So that is the apoenzyme. So this binds to a cofactor to form the whole enzyme, which is called the holoenzyme in such conditions. So in general, the properties of enzyme and enzyme catalysis is that the enzyme is required only in small concentration, and the enzyme accelerates the rate of the reaction by lowering the activation energy, and thus reducing the activation energy increases the number of reactant molecules that can overcome the energy barrier and get converted into the product. And so the enzyme catalysis does not alter the physical or chemical properties of the end product, and the properties of the enzyme itself is not changed. It remains unchanged by the end of the reaction. And so enzyme catalysis also accelerates forward or reverse reactions to attain equilibrium. That is, the enzymes catalyze the reactions in both forward and reverse directions. And like you said, the enzyme remains unchanged at the end of the reaction. And the enzymes sometimes require hydration for their activity. So this activation energy is also called free energy. So free energy is nothing but the difference in energies between the substrate and the product. And it's called the change in Gibbs free energy, that is delta G. So also delta G indicates whether a reaction is thermodynamically favorable or not. So a negative delta G value indicates that the reaction is energetically favorable, and a positive delta G indicates that the reaction is not favorable, and delta G is independent of the rate of the reaction. If delta G is small, it doesn't mean the reaction might happen faster or slower. So these are just independent terms. And moreover, there are several theories explaining the mechanisms of enzyme catalysis. And before that, let's look into two hypotheses that tell us how the enzyme interact with the substrate. So one is the lock and key hypothesis. So here the structure or the conformation of the enzyme is rigid or unchanged. So the reactant molecule or the substrate, which has the opposite charge to the enzyme, fits in the surface like a lock and key. So thus the active site of the enzyme is rigid and pre-shaped template where only a specific substrate can bind. The other one is the induced fit model hypothesis. So here the active site is not rigid and it's not pre-shaped. So the enzyme is flexible. So the essential features of the substrate binding site are present at the nascent active site. So the interaction of the substrate with the enzyme induces a fit or a conformation change in the enzyme, resulting in the formation of a strong substrate binding site. So further, due to induced fit, the appropriate amino acids of the enzyme are repositioned to form the active site and bring about the catalysis. So after the substrate and the enzyme bind together or combine, mechanisms of catalysis lower the energy of the transition state, which is done by forming an alternate pathway for the reaction. So there are several mechanisms and theories of these alternate routes that explain the mechanisms of enzyme catalysis. There's substrate strain theory. So the substrate is strained due to the induced conformation change of the enzyme. So the substrate changes its conformation so that it can combine or bind to the enzyme. So it is also possible that when a substrate binds to the preformed active site, which is like the lock and key model, the enzyme doesn't change its conformation, but the substrate has to change its conformation to fit into the enzyme. So when the substrate binds to such preformed active site, the enzyme induces a strain to the substrate. So the strained substrate leads to the formation of the product, like you can see in this image. So a combination of the induced fit model with substrate strain is considered to be operative in the enzymatic action. And then proximity catalysis or propinquity effect. So this effect theorizes that the proximity and the orientation of the enzyme substrate complex align the reactive chemical groups and bind them in an optimal orientation and spatial relationship for the reaction to be successful. So the rate of the reaction increases because of this effect, and it is similar to the effect that occurs when the concentration of reagents increase and thus the rate of the reaction increases. So it's just that the enzyme and all the reactant molecules, they are close together and they are in such a conformation that their reactive groups are closer and they can easily react to form the product. For example, like to have a simple explanation, you can see that A and B are the reactants here, like it could be the enzyme or the substrate. So they are in a specific orientation where the reactive groups are not close to each other. So here the rate of the reaction is much slower. It's like very slow. So whereas here both the reactants, they are in a certain conformation that brings them close together. So this reaction is like 200,000 fold faster than it would be when they are not closer to each other. And here orientation effects, for example, like as compared to this orientation, if the enzyme and the substrate are in this specific orientation, the product is just formed faster. So this explains the proximity or the propinquity effect for enzyme catalysis. And moreover, during the reaction, several ions are exchanged and intermediate unstable molecules are formed, and this state is called the transition state. So, enzymes help stabilize the transition state by exchanging ions among different molecules or just by holding the ion in place until the reactants meet them. So, explaining such mechanisms, we have acid base catalysis, and this is a catalytic reaction where protons, that is hydrogen atoms, are exchanged in the presence of weak acids or bases that donate or accept the protons. So these act as the enzymes in this case. So basically acid base catalysis is a transfer of protons mediated by an acidic or basic enzyme. So to stabilize the charges that are being formed in the transition state, the proton donors or acceptors, which are the acids or the bases, may donate or accept protons. So at the physiological pH, histidine, the amino acid, so this is the most important amino acid. The protonated form functions as an acid and its corresponding conjugate performs as a base. So here's a simple example of a reaction that undergoes acid-base catalysis. So ketone is converted to enol, and the acid catalyst here acts as an enzyme. It donates a hydrogen ion to stabilize the negative charge delocalized on the carbon atom of the unstable carbanion formed during the transition state. So in simple terms, so this catalyst, it donates the proton, it donates the hydrogen ion to this O group to neutralize its negative charge. So it forms COH here. Whereas the hydrogen atom at this side chain, this is being removed from the reactant, and now this intermediate enzyme, it accepts the hydrogen atom from this group. So now we have our enzyme back in place, and we also have the product formed. So here it's just the movement of hydrogen atoms. So the hydrogen that was here is now here, and that was facilitated by the proton that came from the acid catalyst. Another mechanism is metal ion catalysis. So this theory states that the metal ion present in the active site engages in the catalysis through the coordination of charge stabilization and shielding. So since the metal ion is generally positively charged, only the negatively charged ions could be stabilized, and pH does not affect them, and hence it is advantageous in catalysis reactions, whereas in general catalysis is favorable in a certain pH, in the physiological pH, and at a certain temperature. So metal ion catalysis is not affected by pH, so that's one of the advantages. So in this case, like we just saw, the metal ion participates in oxidation reduction reactions, which involves the movement of electrons. So the metal ions may interact directly with the substrate, or they may just stabilize the charge of the transition state, like in this example here. So here the zinc metal ion neutralizes the negative charge of the carbanion in the transition state, and it just waits for two hydrogen atoms to be received from the water molecule for it to form a product, which is ethanol in this case. So this negative ion is stabilized, and it receives one hydrogen atom, is forming OH, an ethanol alcohol group, and another hydrogen atom, which we see here. So this is the mechanism of metal ion catalysis. Another mechanism is covalent catalysis. So this is the process of lowering energy of the transition state by covalently binding to the side chain of the reactants. So here the enzyme cycles between two states. So at first it covalently binds with the reactant or the substrate, and then during the later stage of the reaction, this covalent bond is broken to reproduce the enzymes. This process does not lower the activation energy of the pathway, but instead it provides an alternate pathway. So here's an example of covalent catalysis where acetoacetate is decarboxylated to form acetone, and enolate is the unstable intermediate. The arrows here just indicate the direction of the flow of the ions from one atom to the other. Now let's get into a brief overview of the details of the reaction we just saw. So here a primary amine acts as the catalyst. So R could be any side chain and NH2 is the amino terminal. So RNH2 reacts with carbonyl COO minus to form imine, which is C double bond N. So here RNH plus ion is covalently bound to the substrate and hydroxide is released. Here, the protonated nitrogen atom reduces enolate of the intermediate. So the COO minus here loses an electron and is released as CO2. The intermediate then accepts a proton and the CH2 group becomes a CH3, while the enzyme RNH plus is covalently bound to the intermediate. The intermediate, which is a Schiff's base, now again reacts with the hydroxide, forming acetone, which is the end product, and the amine catalyst is regenerated. So here C double bond O is formed, and the protonated RNH reacts with hydrogen from the hydroxide and forms RNH2. So this is just one example of covalent catalysis reaction. So here just the side groups are just displaced, they're replaced, while the enzyme just acts as an intermediate and helps in the transfer of the ions amongst each other. So far we have seen some of the basic mechanisms involved in enzyme catalysis and the properties of enzyme catalysis. So now coming back to a question, identify the statement that is not applicable to an enzyme catalyzed reaction. So option A, enzyme catalysis involves propinquity effects. So we saw that proximity or propinquity effect, it helps in increasing the rate of the reaction because closer the reactants, the reacting groups of the enzyme and the reactants are with each other, the faster the reaction occurs. So this is this is not the answer to the question because this is correct about enzyme catalysis. Then option B, the binding of substrate to the active site causes a strain in the substrate. So we saw the substrate strain theory, and so because of the induced fit model, the substrate undergoes a change in its conformation to fit into the enzyme's active site, and so this causes a strain in the substrate which leads to the catalysis and hence the product formation. So this is correct about enzyme catalysis, and so it is not the correct answer to the question. So whereas option C, enzymes do not accelerate the rate of the reverse reaction. So this is false because we saw that enzymes mediate or catalyze the reactions in both the directions, both forward and reverse directions. So this option would answer the question. So this is not applicable to an enzyme catalyzed reaction. So this is the answer to our question. So the other option that's left is enzyme catalysis involves acid-base chemistry. So we just saw acid-base chemistry, which is one of the mechanisms of enzyme catalysis where transfer of protons, that is the hydrogen ion, occurs. So this is correct about enzyme catalysis as well. So the statement that's not applicable to enzyme catalyzed reaction is enzymes do not accelerate the rate of reverse reaction.